Well, hello, Misfits, and we're here with Mat Seidensticker from,
Northern Rockies Research and Educational Services.
which means he is super in the moths. We can all gather that from that name. So Matt has been a friend for a super long time,
Yep,
and he started off as a owl guy,
that's right.
and then an entrepreneur, and then a really bad bartender. And then back going into the sciences again. So we'll talk a little bit about the night shift. It seems like you have this whole arc of your career of working at night.
Yeah, that's very true. Much to the to the demise of my wife, of course, right? But yeah, you're right. I mean, I started working with the Owl Research Institute when I was out of undergrad, and I worked with them for about a decade. And so obviously, owls, so plenty of night work there. And like you'd mentioned, now I'm chasing moths around. So yeah, I just can't seem to kick the nocturnal habit, if you will.
So what's next? Bats, night jars.
That's right. Well, we've had some of that, with my work in MPG. I've had plenty of that going on too. We can talk a little bit about that.
Yeah.
That's sort of how this moth thing got started, in fact.
So let's kind of roll back to the beginning, and we'll start at the beginning. We'll roll back to the beginning. And so you actually started out young Matt, bright-eyed, eager. You played college basketball for the Montana Grizzlies.
That's right, I did. Yeah, you're making me feel old now.
Oh, this is back when they shot the free throws underhand?
Yeah, almost.
So how do you, because you don't see a lot of folks who are involved in athletics go into the biological sciences.
Right.
So first of all, what kind of made you gravitate towards the biologic sciences?
I think, Rad, this being a small town Montana kid, that it was just my upbringing, just being in rural Montana. And I came from a family that was, really into hunting and things like that, and a little bit of a ranching background as well. And so, I just spent a lot of time as a child outdoors. And so naturally, I just had this kind of innate interest in animals and the outdoors. And yeah, and then once I got to college here in Missoula, I mean, obviously our program here is like top notch, and so I just I was a kid when I was younger. I had I still have them. In fact, I tried to pass them off to my now 13-year-old son, which he's like, "What are these, Dad?" But I used to have this subscription where we call them the safari cards. I called them, right?
Oh yeah,
where you get
those.
the little pack of cards, and they'd have an animal, and on the back they'd have range maps, and so. It was just always kind of something I was interested in, and it just kind of carried through into my older years. Yeah, yeah.
And so you just, when you got to university, you're like, oh, that's the direction I'm going to go.
Yeah, exactly. And part of it too, my dad was a science teacher in high school, and so when I first started out in college, I was actually a zoology major. So I was kind of interested more in the molecular end, and maybe even trying to pursue something in medicine sort of thing. But midway through my college, I switched to wildlife biology just because I realized that I wanted to be outside. I didn't want to be in a lab necessarily, or looking at small things. Although now it's kind of
we'll get back to looking at very small things.
No, it is. But so yeah, yeah.
So when you got done playing and now you're graduated, the hardest thing about, and I can speak from experience on this too, is as a wildlife graduate, finding a gig in your field. I mean, you can find a gig washing dishes, but I mean, to actually find a job in biology can be really super tough.
Yeah.
And did you make the jump right to the Owl Research Institute?
I did, yeah, and I was pretty lucky. Speaking of being a bartender or washing dishes or whatever, in fact, I met Denver at the depot when I was bartending there, and he used to always come in, right? one of the waitresses had kind of, told him that, hey, this guy that, he's graduating, whatever, he's really interested in wildlife and stuff like that. And so, I met Denver kind of indirectly through there, and he invited me out into the field, sort of a, kind of a test run, if you will, or whatever, to see what, and then it just kind of snowballed from there. So I was really lucky, to jump right in. And I got to go to Barrow that first summer with him. And I started out as sort of like.
Now, when you say Barrow, you mean Barrow, Alaska.
Barrow, Alaska, right?
And you're definitely doing snowy owl stuff.
Definitely doing snowy owl stuff up there. And so, it kind of started out as like a sort of an internship sort of level role. But then eventually after that the first summer, it evolved into that. And it still wasn't necessarily like full time for a lot of those years. But like you said, we often joke in Missoula, especially here, right? We've got more PhDs and lawyers washing dishes and working at the coffee shop than anywhere in the world.
You're surprised where they're working at, yeah.
So I was really lucky that way.
And so you started off with Denver doing the Snowy Owl Project, and then did you move on to these other projects, doing things like saw-whet owls and flams? And
I did, yeah. So I worked on the Snowy Owl Project for 7 summers, but in the off season of that, I would work on the long-eared owls. I was fortunate enough to be involved in some of the northern hawk owl research up in Glacier Park, and then. Here and there we do little stuff like pygmy owls. Those projects were, as some of those species are really hard to find. So they were really sort of limited time windows, throughout the year. But so it was mainly long-eareds and snowies, and then eventually I went to grad school where I evolved into the flammulated owl research.
Right.
And so that took place about 10 years ago or so.
Now, flammulated owls are my favorite.
Yeah.
Because one, they're teeny tiny.
Yep.
They're super nocturnal.
Yep.
They have all dark eyes. And they have a unique characteristic on what they choose to feed upon.
That's right. Which is a great segue into
what you're doing
what
now.
I'm doing now, right? And so that is when I kind of, as Rad was mentioning, flammulated owls eat mainly moths. They're well known as kind of insectivorous owls. And so that's where my interest really started with moths, was during my graduate program. And then once I got done with that, I moved on to working with the MPG Ranch, which is, a local conservation ranch here in the Bitterroot Valley. And we started a project. We wanted to know what these nocturnal insectivores were eating. So like flammulated owls was one. We knew that they ate a lot of like moths, but we also wanted to, we knew that they eat other stuff too, right?
Now here's the question. When you say flammulated owls eat moths, how are they eating moths? 'Cause I have this two things. I don't have a ton of time with flams, maybe 12 hours total on territory with flams.
Right.
But it seems to me, are they picking moths off of limbs, or are they picking them out of the air?
I think predominantly they're sort of hover gleaning them, yeah. So they're sort of, a lot of these moths, especially like the spruce budworms, some of these smaller moths that are associated with pine, they sort of gather around the needles and the branches of the pine trees. And I think a lot of times that's what we've observed it before, where they're sort of, they're just kind of just hovering around those tree limbs and stuff and just picking them off. Just
picking them off as they land there to do sexy stuff with each other.
Right. And some of the larger moths, you see pictures of them with like a big like polyphemus moth or something like that. Those, whether they're getting those out of the air or not, I'm not sure. I'm sure it's pretty easy for them just to grab them with their talons and switch it to the beak or whatever. But I think for the most part, it's sort of a hover gleaning kind of situation,
which is really kind of cool for an owl to be doing. To be really behaving.
Cool. Really neat. And when you watch them fly at night, they're easily mistaken for like a bat. Really. Yeah. They have these sort of long, slender wings and they have kind of rapid wing beats. They often look like a bat, yeah.
Right. And the other thing that I think is interesting is how cryptic they are.
Oh, yeah.
Like, I remember, like, when, not Denver, who was, oh, God, my brain totally leaves me. The gentleman from the Forest Service who had some of the first ones up Patty Canyon. Do you remember his name?
Yeah, you're slipping my mind.
But it was outside of Missoula,
Yeah,
and he found, like, it was like this big groundbreaking thing. Like, oh my, there's this pea pine kind of associated species all the way up here in Montana.
Right.
How cool is this? And then you start finding them. Well, if we look here at night, we found them over here.
Right.
We found them around the valley.
Right.
And then Helena, they start finding a bunch. And now all the way out at Lewiston, they're finding them. And
and
they're not that rare.
No, they're not. It's the classic thing of just, people just never went out and looked for them, really.
Mm-hmm.
Right. And so that's definitely the feeling we got was, like you said, they're sort of associated with those open pine forests with low understory. And if you go out to any of these ridges in western Montana that have, larger trees and open, you're going to find plenty. I mean, one of the big things that's limiting them now that I think is they're obligate secondary cavity nesters. And so one of the things we found in our study is that
we should probably stop and tell the people in Plains what obligate secondary cavity nester is.
So that just basically means they don't build their own nests. So they use like old woodpecker cavities,
flicker holes, pileated
holes. Woodpeckers, exactly,
right?
Or old hollowed out, branches or whatever that have decayed out. And so I think that they're probably limited more by available cavities than anything, because I think the traditional forest management, right, was like, let's take down all the snags and let's get rid of this stuff. And so I know in our study area, we were up in Marshall Canyon, and it seemed that there was not a great density of snags, and available things. So but anywhere that you have snags and you have an open forest, you're going to find.
Well, I know one particular little spot on a little ridge that's a home run, because there's like 4 or 5 huge snags there. And
and
they're out there every year.
They're there every year. And the other thing, like you mentioned, how cryptic they are. So finding their nest is like really, really difficult. It's they're really nocturnal, like you said, and they're very, very secretive. And so, basically what you have to do is we'd have what we called a peeper camera, right? So it was like a telescoping pole with a little camera on it that you could stick in cavities or whatever. And so we would inspect a lot of cavities, but the other thing is that not all cavities are created equal, right? So you'll see like a really nice, and apparently flams, they're a small owl that like a big cavity. So
they want it really going down deep.
They want it deep, and they want a large entrance hole, even though they're so small, versus like a pygmy owl, right? That'll squeeze in like a
hairy woodpecker hole.
Yeah, like a 1-inch hole, and So, and they like to nest really high up, these flams, and so. But the interesting thing we found is we surveyed, I think, 60-some trees with over 150 cavities, and not all cavities. So you'll stick that peeper camera in a cavity, and it looks great from the outside. It's got a 3-inch hole, nice and round, beautiful. You stick it in, and it might be only like 2 inches deep.
Right. Which is barely excavated, more of a foraging hole.
Or
they quit on it.
Or you'll look in there and it's got good depth, but the floor is all decayed and spiky, or there's no floor at all, you know. And so it's they get limited in that regard, but they're abundant.
There. And so when you say that they're looking for these particular holes, I know, and I'm not encouraging people to do this. I may have done this on a few occasions. But every once in a while, I'll take a stick and tap against a snag, and every once in a while, more often than not, a flying squirrel will poke its head out. Every once in a while, northern pygmy. And a couple times I've had a boreal poke its head out. Do the flams just not react that way? They don't poke their heads out and take a look?
We've had mixed results. I mean, we've definitely had, like we were going to go to ban the nestlings, and we were roughing around down up below the tree. They would stick their heads out. Right. And we've also had results of banging on a tree and having them stick their heads out. I think part of it with flams is just the height that they nest in.
So even if they poke their head out, it's a little tiny.
You might not even notice it. You might not even notice it. I mean, we had one nest that was 72 feet up. And it was in like this, like near the top of this big pea pine, and it wasn't even like necessarily a woodpecker hole. It was more of like an old branch that had broken off and then sort of decayed, and it was shielded by like another branch that sort of came out in front of it. And so it took Matt Larson, my colleague, like I can't tell you, countless hours of like observation. And so basically that's what you have to do is if you suspect a cavity, you'll go out and just watch for feeding observations. Them, the males will come and bring food, and so you just have to sit there and watch and watch and watch. And then when it's that high up, you're like, okay, what, where did it go? Like,
right.
what cavity was it? Things like
which
that.
Cavity was it? Because this tree has about 80 of them.
Yeah, right. And so, but finally he nailed it down. So they're really hard. I think we only found like in our 3 years of research, like 4 active nests.
Right. So
one was reused 2 years too.
So there is a reuse.
There can be, yeah. And flams are really they're known to like come back. They show fidelity to like their breeding territories, so they'll actually. And we had a couple recaptures where we banded, and they caught them in the same territory the next year.
And this is very this is very fecal. But do flams do fecal sacs? Then they take them away and drop them because they're not whitewashing the nest hole.
No, yeah, and that's interesting you say that because the cavities we did. We're able to access and like get the nestlings and stuff. They're very, very clean.
So they're
obviously
when they leave the nest the first trip, they're taking out the garbage.
Yep, exactly. And that's probably smart, right? Because they, they're nesting in these cavities with big entrance holes that could easily be accessed by, martins or squirrels or whatever and stuff. So,
well, and that leads me to the next question. It's kind of like when you bring up predators. What did you see in nest depredation, or was the sample size so small that you never was observed? Or
we never observed it in our study, and actually, Linkhart in Colorado, he's sort of he's been studying them for like 35 years or something down there, and he hasn't observed a ton of instances either. It's mainly been like you'll find like. He found an adult female in a cavity with like missing a leg and, part of its head. It was suspected like squirrels or something, they'll just kind of go in there and rampage, right? Just kill stuff to kill stuff basically. So, I mean, some people say that, great horned owls maybe, sort of, but we actually had, great horned owls and flamingo owls calling in the same territories in the same nights, so.
Well, isn't there a segregation? I mean, what would be the competition? I mean, you have an invertebrate versus vertebrate.
Yeah, right.
Segregation.
Accipiters, I think, have been implicated in a couple adult mortalities.
Well, sure, if they catch them flying.
Yeah, if they catch them flying or whatever, or if they spot them at a roost during the day or something, they might just take them.
Yeah, go ahead.
They really, I think it's pretty rare. At least documented.
That's rare.
It's rare. So
you worked flamulateds for how many years?
That was from 2008 to 2011, so 3 years.
So 3 years you're working just on flammulated owls.
Just on flammulated owls.
And this is all around Missoula.
It was. Our primary study area was up Marshall Canyon and the Woods Gulch area. That's kind of a hotbed. I mean,
not that everybody knows.
Not that everybody already knows, right? But then we also hit some historical areas from when the Avian Science Center at the university did that statewide study.
Now it really kills me. I can't remember that guy's name.
Yeah, well, there was Amy Sillenberg was running at the time, and
then the other Denver. The other Denver was involved,
right?
And then,
of course, Dr. Hutto was running the show at that time.
Yeah, Hutto, and then
there was Christina Smucker, I think, was involved in that.
And wasn't Dial doing a little bit of stuff with it, just tangentially, or was he just still on his acoustics and flight thing?
Yeah, I think he more was into the acoustics and flight stuff, not so much the field work. But so they did like a whole statewide. I think they surveyed every national forest in Montana, and so we had identified a couple areas. There was one, over by out by Clinton,
Mm-hmm.
and then some up in the 9 Mile area. So, pretty good hotbeds for them. So, but, as a grad student, right, we were a little cash strapped. So it was nice having, just right out our back door.
Well, literally, if you had to ride your bike to it, you could.
You could. Yeah, exactly. You could.
At night.
At night, doing surveys with a Fox Pro or whatever,
Yeah.
So it was nice. And there was such a high density up there that we were kind of able to,
Mm-hmm. And so you work for Denver a long time. And Denver's still going.
Still going. Yeah, as successful as ever.
Yeah, it's I've gotten a chance to go out with him a couple times.
Yeah.
He's the only guy I've ever seen hand catch a saw-whet.
Oh, yeah. Yeah, I don't know.
I saw him hand catch a saw-whet and a long-eared owl.
Yeah. Oh, yeah.
Which is craziness to me.
He's a talented guy. He loves to climb trees and he loves the. Yeah, he's doing well.
And so you're kind of done at ORI, which is Owl Research Institute,
Right.
and then you start a business downtown.
That's right.
And then from there, you go to MPG. Now, when we say MPG, everybody around here kind of knows what it is. But can you explain what MPG is as an entity?
You bet.
It's a little weird.
Yeah. Well, it is a little weird. So it's a private conservation property. It's about 15,000 acres. It's down kind of east of Florence, Montana. In the
Bitterroot Valley of Montana.
In the Bitterroot Valley. And it was an old cattle ranch, and it's basically kind of set up as like a research field station in essence. So a lot of restoration goes on. And just a lot of just pure like research, everything from like soil microbes on up to big game. And I really can't tell you what like the end game is or why it was set up necessarily. But all I know is it's been such an invaluable resource to a lot of like biologists around the area.
Right.
I mean, there's just so much going on and so many opportunities and to get out there, and to be able to do this kind of really, really neat research. So I started working for their bird crew basically in 2015.
So you're working for Kate, right?
Kate Stone, right? Yep, she heads that up, and they were just starting projects on common poor wills, which is a nocturnal little nightjar,
right?
As you know,
right? Which is another bird, just like flam. Everyone thinks they're super duper rare. Turns out. Not
so much. In fact, I used to travel around the state, and we'll talk about this more, but to do moth stuff, and I was hearing them just about everywhere I went last year in eastern Montana and south central Montana. They're all over, they're all over the place. But for western Montana, it was really like, and we even detected them on our flam surveys.
Right.
And during my graduate work. But so it was
I think on right side of Missoula, like Crazy Canyon.
Exactly. Yep, that's where we had them in our flam surveys, right? Yeah, and then up Marshall Canyon as well. But for western Montana, it turned out, MPG started kind of doing some surveys, and it turned out that like MPG was like kind of a hot spot for them, and so they that was one of the first years they started kind of doing surveys and investigating. We did some telemetry work. Looking at, like roost site characteristics and just, breeding phenology and some basic like natural history stuff.
Right.
And so did that for about a year or two. And then in about 2017, we were like, well, geez, what are these things eating? Right. And then about that same time, in 2017, they also got interested in nighthawks, common nighthawks. Right. And then Nate Schwab, who works at Tetra Tech, and he was, got his degree at the university. He's a bat expert. He was also starting some bat work on the ranch, and so we were like, "Geez, what are these things eating?" Right? I mean, we know of like there's been studies right from stomach contents or people dissecting the poop to look for like insect parts and stuff.
You're looking at carapaces,
stuff like that, right?
Which only gets you down to
like order level, maybe family, right?
Yeah.
And so it was around that time, too, that we discovered this molecular technique called DNA barcoding. And so, it's basically barcoding is it's basically just like sequencing a really small part of a gene region that it's. These gene regions are so highly conserved within species, but variable enough between species that you can tell, like you can take a tissue sample or whatever and sequence it and then compare it to like a reference barcode and be like, okay, that's what this species is, or that's not what animal this came from.
So like for mushrooms, people do ITS?
ITS, yeah, exactly. Right. And then so with with animals, it's the CO1. Yeah, which is a mitochondrial gene.
And so you guys are literally were you taking fecal matter and then
Exactly. So not only can you use it to identify species, but you can use the technique to amplify DNA in fecal matter.
Right. So it does and this is the amazing part, but it takes all these gene regions and knows what they are and then magnifies them. Right.
And then you sequence them out, amplify the DNA, and then you can compare those sequences to reference sequences in these reference databases. And
like GenBank?
GenBank's one of them. The one that we work...
Or GenBank.
GenBank, yeah. The one we started working with is called BOLD, so it's the Barcode of Life database. And it's... and
do they share data?
They do.
Those two?
Yeah, they do, yeah. And so the BOLD program is out of the University of Guelph in Ontario. And in fact, Dr. Paul Hebert, he's sort of considered like the father of DNA barcoding. He started kind of developing this stuff. And so their database is really cool because they're all connected to voucher specimens. So we're like GenBank, it's just like a DNA sequence and nobody really knows where it came from or if there's even a voucher insect that it's tied to or whatever. BOLD,
right. It's more open source.
Right, so it's more prone to misidentifications or that's not easily verified sometimes. And so BOLD, they require you to like take a picture and submit all this metadata and all that stuff, so that you can link the sequence to the animal, which is cool. Well, so we're like, oh wow, the light bulb went off. So let's nighthawks and poor wills. I mean, they leave behind these like really distinct like fecal pellets at their roosts, right? And you can just collect them up.
Gather them up.
You just collect them up by the dozens, and so and they're really
gathering the poo,
right? and sometimes like obviously they'll do it in your hand or whatever too, but the cool thing about this DNA barcoding is it can work on older or things that have been exposed to the environment, right? So like once the insect DNA is kind of in there, as long as it's not like highly degraded from like rain or like swings in high heat and stuff like that, it's pretty easily preserved, and you have good success. And so we were like, okay, let's just start collecting poop and let's have it like sequenced and see what these things are eating. Well, we quickly learned.
So when you guys collect the poop, are you going through and like macerating this shit and starting to magnify, or are you sending it off to somewhere else?
We just send it right to BOLD. So they
do all the PCR and
everything, which was slick because it's so the BOLD database. The main purveyor of that is the Canadian Center for DNA Barcoding, which is based at the University of Guelph as well. And so they were great. Yeah, when we just communicated with them, and so we would just basically once we collected the fecal pellets, we just put them in like a tube with some ethanol or whatever to preserve them. And then at the end of the year, we would just ship them all to the lab and they'd do all the DNA extraction, sequencing and everything. So, so that was really cool. I know there, a lot of studies, you can get these like DNA extraction kits that you just buy over you can just do it yourself and then you just send the DNA to them or whatever.
You'll love it. I know a fungus guy.
Yeah.
Who has a PCR lab in the backseat of his car.
Yeah. Well, and that's where it's
and he's literally getting them to like, oh, I got good DNA on this fungus. Now, to actually do the analysis, he sends it off.
Exactly.
And it's like $8 a sample or something.
Yeah, exactly. And see, and that's where you have to weigh the cost effectiveness. So those kits are, we kind of broke down the numbers. And so we were like, well, geez. And MPG certainly has the capability to do it in-house. But by the time we kind of like looked at what it would cost to just send it to the lab and have them do it all versus buying those kits and doing it ourselves and the time and all that,
right?
It was like in fact, I think we were saving a little money.
Yeah, you're going to spend $16. Go ahead.
Yeah, so we were just like, let's just let you do it. We need to focus on finding these birds and the pellets. Well, one of the things we quickly learned is even though this technique is successful at amplifying the DNA and stuff like that, and that's not a problem, right? I mean, that's pretty straightforward and been well established, is that even though like Bold, right, has like, I don't even know how many million sequences or whatever in their database, the taxonomic coverage of species in the database is still relatively lacking, especially for insects, right? Where we're talking like 1 million described, maybe up to 100 million that exist, something
and
like
you
that.
got these weird clusters that have like a lot of data, but it's a very narrow genera.
Right, very short sequences that you're actually amplifying. And so like the full barcode for CO1 is like 650 some base pairs, right? But the actual stuff that they're amplifying out of the poop is only like, you're lucky if you get maybe 200. So it's just a fragment of that barcode kind of thing. And so you really need to have like a solid reference sequence, right? Or else you're not going to get somewhere. So we kind of realized that, especially for insects. So we were like, another light bulb went off.
So you're literally getting these reference, and you can get it down to, and I know that this is so old fashioned because things have become so much more complicated with phylogenetics and everything else that, okay, now we have this complete realistic picture of how things evolve and how they're grouped. But it's no longer in our nice, neat, order, family, genus. That's all broken apart. And now those have become literally ad hoc. We just place those where we can group a large number of things. And we'll call it an order, but on the level of things. So you're literally finding Montana, like, okay, we can get them to this big group, but we don't have any specificity down below. Right.
And so it's pretty reliable to genus.
Right.
I mean, most of the sequences that you get, you can pretty much count on like the solid to genus, but there are a lot, right, that only come down to family, because sometimes there's sequences in like the database, and even though it's tied to a voucher, maybe that specimen hasn't like necessarily been identified by a taxonomic expert.
Right.
And so when it hits on that database or on that sequence in the database, it'll only go to like what taxonomy that's already been assigned, and so, or it just might not be in the database. And so,
then how many of those groups, especially with bugs, because it's so, one, the coverage is so small. Two, there's so much work to do. How many of these genus, orders, families, all the way up, are you finding that are polyphyletic and they're not really real? Like, we kind of lumped all these things together, but it's not real.
Right. Well, and that goes like, I mean, you do have instances, and I've learned this with moths, is they'll like numerous species will actually share traits of the barcode, right? So it's like, so you're like, can you really tell what is what? You know what I mean? And that's an issue as well, for sure.
Right. What's well?
Well, what is it actually even its own? Is it a unique species?
Right.
Or is this all the same thing, and it's just got a bunch of variability in its genes and
or in the process of going their separate ways, but they're not quite there yet.
Right. Yeah, exactly. And we haven't looked too heavy into that. Although we do have a data set that we might be able to do that, but so we are kind of like, oh my God. So if the results of these fecal cell, if the results from our fecal DNA are only as good as like the reference libraries we're going to compare it to, and these libraries are lacking taxonomic coverage, we were like, well, why don't we make our own library? Why don't we start adding to the reference? Right. Why don't we create our own local library? And not only will it help, build a taxonomic coverage in the Bold database, but it will give us sort of this extra layer of corroboration, because these are the taxa that we know were occurring in the area at the time we were collecting, these fecal samples. So it kind of, gives you a little more like, okay, well, yeah. When you get the DNA, you're like, yeah, that's probably what it was eating, right? Because we caught it in the study area, and it came back solid DNA hit and all that stuff. So we, in 2017 and 18, we started just in earnest, started collecting nocturnal insects. And I was using a variety of techniques. I started using light sheets, which is a common mothing thing where you hang up a white sheet and you put a light in front of it. And
UV light. And
like a mercury vapor light and a black light. And then the moths come to the sheet, like a moth of the flame thing. And so we started, and then we used Malaise traps too, which are, pretty standard, just like net, traps that have a collection bottle on them, and, insects. And so we started using a variety of those things to collect, to build our own sort of like local reference library. And that's where this moth thing kind of just went off. I think the first year in 2017, we barcoded like 320 some species of moths just on the MPG ranch.
So you're talking this place is huge.
Right. Now
you're covering all 15,000 acres?
Not necessarily. So the first year we were just doing basically in the areas where we were trapping the nighthawks and the poor wills. But that did cover, a lot of the mid elevations like sage steppe sort of, conifer transition
all
zone.
the way up to like
all the way up into the, coniferous forest. And I think that first year we had maybe 6 or 7 sites that we did like once a month from May, June, July, and August.
Okay.
So we would go out and one night we would sample, we would light sheet in those areas or whatever, and I'd hang up like an aerial malaise or whatever, and stuff. And so that first year, it was like 300. And so we were just like, holy crap, like what? Like, wow, that's a lot of moths for this little ranch, And then the next year we went and we added, different sites in different areas and maybe a little more geographic coverage on the ranch. And pretty soon, we were up to like 500 species of moths on the ranch, And then at that point, after 2018, I sort of started kind of wondering, like, well, geez, like what is going on with moths in Montana, right? I mean, if we have 500 species, just in not like a great amount of effort,
right?
What is there any research going on with moths in Montana? What's going on? And so I really found out that there wasn't much, besides there's a fellow named Bob Martin over in Helena, and he's been mothing in his backyard for like 20 years or something, right? And so he's working with like the LEP Society and stuff and submitting observations. And so I kind of got a hold of him and he's like, oh yeah, I've photoed, and he doesn't collect it all or anything. He just photographs them, but
photographs them on the sheet and
then just
they go on their merry way.
And so he was like, geez, yeah, I've some like 1,600 species over 20 years, just in my little area in Helena, and I'm like, holy crap, this is like amazing, right? And so then, part of this, like, this was like iNaturalist, and then there's this app called LEPS. It's like a AI thing that you can submit moths to and stuff. So we just, as part of our kind of like, I don't know, due diligence or whatever with our MPG data, we just started submitting pictures to like iNaturalist and LEPS and stuff. And well, in 2019, I was contacted by a gentleman who was real interested in one of our records on the ranch. And his name is Chuck Harp. And Chuck is the Rocky Mountain coordinator of the Lepidopterist Society. And he's also a research associate at the C.P. Gillette Museum of Arthropod Diversity in Colorado, at Fort Collins, Colorado State. And so he kind of reached out and was like, hey, can I get this record? I want to include it in my report, at the end of the year and stuff. And then one thing kind of led to another. We had a couple phone conversations, and I sort of grilled him about, like, because he actually lived in Great Falls and Billings for a time. He was in retail for his whole career, but he's a moth expert. He manages Montgomery.
Well, there's not a lot of money in moths.
No, there's not. There's not a lot of money. And these other guys, it's funny too, because like Lars Crabo, who he's like described like new species and this and that. And he's a radiologist, right?
Right.
And there's all these these are serious, like, experts in the field, and they just love doing it. So anyway, I started asking Chuck, like, well, geez, what's been done with, moths? He's like, very little. He said, Montana's been historically an under-collected state.
Right.
Given the vastness of the state and all the habitats, who knows? We don't really know. And so then the light bulb went off again. And at that time, too, I was sort of like, that's when I started Northern Rockies, was in 2019. Because I saw sort of this opportunity to do some research on moths and insects in particular.
Right.
And it was about that time where, the insect Armageddon talk was happening and things like that. And so I said, you know what, that's what I'm going to do. I'm going to start this organization, and we're going to, we're going to start it with sort of a mission of like advancing scientific research on insects and moths in particular. And so one thing led to another with Chuck, and the Montana Moth Project was born. And that's our flagship program. And, the overarching goal, I guess you could say, would be, we want.
So you're still working with MPG as this?
And I do. So that's part of there's layers of research. So we don't just do moths. I have a biennial insect biomass and abundance study that I do on MPG. So every other year we have 18 sites that I set up, malaise traps and pitfall traps and pan traps. And then we're just sort of monitoring the abundance and biomass at those sites. And then I also have, as part of the Montana Moth Project, I have a long-term moth monitoring program on the ranch as well. So I have 4 sites that I set a light trap, and we do them every week from April through October. And again, it's just looking at abundance and diversity and how that might change or shift over the long term as like vegetative changes happen on the ranch with the restoration. In relation
which means there was a lot of agricultural fields and that are going back,
and they're going back and doing a lot of plantings and things like that, and then just also in the context of like climate change, like are we going to see like we're trying to, at this point that we're entering our 3rd year, we're starting to get really detailed phenology of like flight times and stuff of these moths, and so
when you say phenology, how discrete is that? Is that all the way down to not daily, but within weekly time spans? Are you going all the way down like the hour? Like, okay, this species is 12 to whatever.
No, it's pretty broad stroke. It's we do it on a sampling on a weekly basis. So, so we're able to get it like, like for some species now we can say, okay, like it's got like a 6-week fly window,
right?
Like, and then on this date it kind of starts, and then maybe it peaks like around this date, and then it tails off, and then it stops, around the middle of July or something, So it's fairly broad. But will we see shifts in those phenologies? As the climate warms.
Are they going to go earlier?
Right, and are we going to see range expansions? Species that may be more southern, are they going to start coming up? And so that's one of the things that's part of the Montana Moth Project on the ranch. And we use non-lethal trapping for that. So we trap live, and then literally, in April, May, sometimes into June, the mornings are still cold enough, and we get out there before sunrise, and so the moths will still be pretty tame. And then we photograph like every single moth in the trap. But when it gets warmer, we actually, we'll take the traps into tents, and you got moths flying everywhere. And then generally what I do is I use, we knock them out a little bit to photograph them. So we'll throw them in a container with a little ethyl acetate for like a minute or two, just to kind of knock them. It doesn't kill them, just knock them out. And then but we photograph everything from the micromoths all the way down to your tiny little, like 1-2 millimeter micromoths. And that's how we get our abundance counts. Because obviously you can imagine, I think last year we photographed like almost 12,000 moths out of our 4 sites over from April to October.
Now, of course, you can't identify all of those at the time of the photograph. So how many of those are...
So that's what I spend most of my winter doing, is identifying moths from photographs, which can be difficult because one thing we're learning too is we're not doing a good enough job of taking photographs of the underwings. So some of these species, you generally photograph moths or identify moths by their maculation on the forewings and stuff.
Right.
But the hind wings are really important too, in terms of, some of them are kind of smoky gray, others are pure white, and then when you start getting into some of these genre that are really similar species, it's the underwing that's going to differentiate it. So, but yeah, that's what I spend my winter doing. We try to keep up with it as much as we can throughout the summer, but.
And are you guys at the same point though with the undercoverage? You guys have to start noticing some of the what's truly a difference and what's truly a variation within species. You
right.
know, that,
Well, and that's the other thing, right? Is like there is a lot of that going on. Different colors, you know,
right.
Like there's a dark morph or a light morph, and then there's,
and you look at the DNA and you go, they're not too different. They're
they're the same thing. And so there's a lot of that going on with some of the taxa for sure, and so over time you work that out, and a lot of times, I'm at a point now where I can be like, yeah, that's a Uzoa, or that's a Caradrina, or that's, this or that. And so you can get it to genus pretty well. And then from there it can be difficult. But for the broader Montana Moth Project, our goal is to sort of better understand the distribution and diversity in Montana as a whole. And how we're doing that is we're collecting and vouchering moths from every county in Montana. And we're working with the Gillette Museum. So we, at this point, after 2 years, I think we've done 22, 23 counties. And that's just one site. Ideally, we want to get 2 sites in each county. But
so it's like a massive survey. It's a broad stroke across the landscape.
It is broad stroke across the landscape, and it's these finite points in time, right? I mean, ideally, we would sample those sites in, like May, June, July, August. Because
and you have multiple sites in multiple elevations, multiple habitats.
Exactly. So we're just starting. I mean, it's a long-term goal. But I think we've sent Chuck, and bless Chuck's heart. I mean, he's a master. He can spread like 300 or 400 miles a day, right? And so he's a machine. So we send all of our specimens. So when we go out to a site, we have traps, and we lethal trap. We put a little ethyl acetate, in the traps and leave them overnight, and then we pack them in freezer like meal prep containers layered in between thick cotton, and it's like moth lasagnas. Right, and we just layer the moths, put a layer of cotton, label them with the site, and then we ship them all to Chuck, and he spreads them, identifies them, and curates them. And I think now we're up to, well, I mean, we're not for sure, but it's approaching like the largest collection of Montana moths in the country, and it actually maybe in North America, maybe even in the world. I mean, we're up to like, I think we've sent him like 18,000 moths over the last 2 years, and I'm
sure that room smells awesome.
God. Well, and it's all housed at the Gillette Museum down there, and so we've actually bought him, I think we're at 4 or 5 24 drawer cabinets.
Just to house all the
moths. And so, this year we're hoping to get maybe 10 or 15 more counties, but we're also just trying to target some of our special habitats in Montana. So last year we got a little grant, some generous support from the Eastern Wildlands chapter of Wild Montana, and we did a bunch of work in the Pryors, which was super awesome.
Which should be really weird.
Yeah. I mean, it was super. I mean, I'd never been to the Pryors until last year, and just that red desert, just really super cool. And so there's a little plant down there called Physaria pachyphylla. It's a little low-growing bladder pod thing, and so there's a gypsum mine that's proposed in that area, and so some folks have petitioned to list that plant as threatened. And so we thought, well, nobody really knows much about the pollination ecology of it. And so, we were like, well, what if moths do? And I guess I should back up that's some of the work that we've been doing too. We've been using this metabarcoding to look at moth pollination, potential pollination interactions by, excising their proboscis and swabbing their thorax.
So you're even getting what they're pollinating.
Potentially, yeah, or at least what the plants they're interacting with. And you can use that DNA barcoding method so you can identify what, like, if you have a pollinium, you can sequence the DNA in it and tell, like, really pretty confidently in a lot of cases, to like genus at least, what plant that pollen came from. Okay. So really cool technique. It's a lot like the,
So now this really ties in the conservation, because now you're going all the way down to the plants. They're going, hey, they need this species.
Yep.
99% of it comes from this one plant.
Right. And moths, are, I mean, in all phases of their life cycle, they're tied to plants. You know, they lay their eggs on plants. The caterpillars feed on plants. A lot of the adults nectar on plants. You know, so every phase of their life cycle is tied to plants, and that's one of the reasons why, in my opinion, they're really great. You know, like indicators, really, of like environmental health. You know, because if you have a really diverse sort of, you know, area of vegetation, you're probably going to have a highly diverse community of moths as well. And there's some moths that, you know, are like generalists, so to speak. So they'll lay their eggs and the larvae will feed on, you know, like hardwood shrubs. It's kind of just like a general category, right? And it could be a lot of different shrubs or a certain family or something, you know. But there's others that are real specific, especially like the flower moths, stuff like that. So anyway, we were like, well, geez, let's see if they're, you know, what about moths? Are moths like interacting with this Physaria at all? And so we went down there. That was part of the project. And then we also were just doing a general inventory. And we collected like 90 moths for pollen sampling. And it was about like 40 species maybe. And we detected the Physaria pollen on 5 specimens from 4 different species.
Okay, so now we're already neck down to like this real narrow band.
Real narrow band. And in fact, one of the species that we detected the pollen on was one that we probably knew we were going to get the pollen because we collected it nectaring on the Physaria.
Right.
It was a little Spilotus, they're almost like a ground-dwelling moth. We rarely see them fly. They're like a groundling moth. They sort of hang out on the ground a lot.
They just walk along?
Yeah, it's kind of weird. I mean, they do fly, you know, and obviously they fly to our sheet and stuff, but a lot of times if you walk around the sheet, you'll see these things that are just kind of on the ground, you know, and this Physaria is a really low cushion plant. So that was really cool. We were like,
that
really
makes me wonder about that, because when you get down there, you have the pallid bats.
Yeah.
Which are odd bats because they hunt on the ground.
Right.
They don't hunt in the air. They actually fly to the ground and
walk. Scurry around. Yeah.
So are they feeding on those moths that are also on the ground?
I'm
everything's
sure.
Low.
I'm sure they probably are. You know, I mean, if they could, I mean, because these things, I mean, like I said, they can fly, but I mean.
And for people that have never seen the habitat, like when you go to the Pryor Mountains, when you're out in these big, just like you said, it's that red desert flats between there and Bighorn Canyon.
Yep.
Anything taller than a foot is kind of an anomaly. It's like like a landmark on the terrain. So everything is super low to the ground.
Yep.
And there's lots of what do they call it? Cryptic soils, you know, that with the living lichen all over the soil. So there's like everything is super low to the ground.
Yeah, it is. It's very, very few like bushes of any kind. You know, I mean, the rabbit brush is about the only thing out there that's of any size. You know,
and it's a landmark. Like from that one to that one,
which is really huge because you know the rabbit brush is. If you want to see moths or catch moths or whatever in the fall, rabbit brush is.
Well, it's someone because you got
the rabbit brush.
You got the rabbit brushes blooming in the fall. Big sagebrush is blooming in the fall.
Yep.
So like you've necked it down to like this.
Just those few species and then you know as far as the general inventory, I mean I think we at this point you know a lot of this stuff is we're still sort of identifying stuff and vouchering stuff, but I mean we pulled at least 12 state records out of there.
Out of the briars.
Out of the briars.
Which makes sense.
Which makes sense. And a lot of these sort of desert, like Great Basin, for example, one of them, this was, and Chuck is a Rhyacionia specialist. So Rhyacionia is a genus of moths that they're called the flower moths. And so they're really like have specific host plants and the adults will kind of rest up in the petals during the day or whatever, and then nectar, and then
You literally mean they're associated with a genus or a species.
Yeah, with a specific,
and that's where they're hanging out.
That's where they hang out. That's where they lay their eggs. That's where they nectar. I mean, it's like they only exist because that like plant is there. And so there's this one called Rhyacionia snowi, which if you look at the moth photographers group, it's like a kind of you know, for Mississippi State, it's like the sort of the go-to for like moth information. Well, previously, and Chuck even said, you know, it was only known from like Colorado, Utah, I think maybe in Nevada a little bit.
But are they like point locations?
Yeah, they just put a point location here or there, and there might be more distribution than what's on those maps. Those maps haven't been updated in a lot, but now we're finding it right in the same sort of habitats in the Pryor's desert,
right?
And that was for a lot of these new records. They were all these sort of desert, Great Basin sort of associated species that we're finding in the Pryors now. And another example is we went to the Centennial Sandhills, the Conservancy property down there by Red Rocks. Super cool area. And so we came up with a dune specialist moth there, Copablepharon, a trinitum that was only known basically from like Mono Lake and southeastern Oregon and some of those dune areas. We found it, and we found it in abundance there. Like, I mean,
everywhere.
I think I collected 160 some specimens of this, and so a lot of it, it's kind of like going back to the flaminate owl thing, right? Like these are all species and taxa that if you look at the range maps and you look at their natural history and information that's what's known, they probably occur in Montana. It's just that there hasn't been
anyone to look.
Right. And so
what is it? Beautiful tiger beetle is on those dunes in the Centennial Valley.
All right. Yeah, that's really awesome. Yeah. And so now we're going to go, like this summer, we're going out to Medicine Lake because that's the only other dune system, in Montana. So we're going to go out there and look for some different Rhyacionia moths because some of the host plants grow out there. And so really neat stuff. I think we're starting to compile our list, and I think in 2 years now we're up to 1,300 or 1,400 probably.
So my biggest thing on all these moth studies, so you have all this, you're doing all this data on moths, there's people doing data on all the nightjars, we talked about flamulateds, we alluded to bats. How does the night shift all allocate across different genera of prey species? So you have all these moths flying. They're doing different things from, like you were talking about that species, that's the groundling moth, all the way to high elevation. How are all these predators, either temporally or layers of altitude, let's call it, how are they segmenting their prey opportunities?
Well, that's interesting. We actually published a paper last year that we kind of looked at that across the suite of the species and how they may be partitioning. And what we kind of determined was, what we found is that there was way fewer eared moths. So moths, noctuid moths in particular, and some sphinx moths, they actually have tympana, right, on their bodies that they can hear. And so there's been this kind of like evolutionary
arms race,
if you will, or back and forth between bats and moths, right?
And they're the classic ones where if they hear the bat echolocating, they literally fold up the wings and just fall out of the sky.
They fall out of the sky, or they'll do some sort of evasive maneuver or whatever. And so we found, compared to like the nighthawks and the owls, of course, and the poor wills, that the bats were eating way less of the eared moths compared to those nocturnal birds. And so we sort of related that to their hunting strategy, right? So, these moths have evolved this like, counter to the bat echolocation, right? Which makes it harder for the bats to, catch them and eat them. Whereas these like, this poor will is sort of like a perch and pop, like almost a fly catching thing, where it uses more of its vision and things like that to sees a moth fly by, just kind of fly catches it, right? And nighthawks similar to.
With nighthawk, I always call them the whales of the sky.
The whales of the sky.
Mouth open, just going through.
Eating the sky plankton, baby, you bet, and so that's one way that they probably are partitioning a little bit, is that, these bats are probably looking, they're honing in more on these like mosquitoes and midges and things that nest candidates here, lacewings, things like that. I mean, they're still eating a fair number of moths, don't get me wrong, but it was by far way more moths in the diet of the birds, right? So so the birds, the nocturnal birds and bats are sort of partitioning that way, perhaps.
So the bats are going more towards like the fly end of the world?
Yeah, like the non-hearing type insects and the the birds are eating more of the hearing aids just because they can ambush them, right?
Right. Well,
they use sight and they ambush them, and the
or mouth open,
and just flying through it.
And just hit my rectile bristle, and I turned my head.
Exactly. And what's really interesting is we found actually the most common prey across all of those taxa was crane flies.
Okay. Makes sense.
Interesting, right?
Yeah,
really cool. I mean, I think
so across all of them, they're saying they're eating the shit. Yeah, all of them are eating the shit out of crane
flies, which is really kind of interesting.
Well, they're big, they're juicy.
They're big, they're juicy. They don't fly very well. A lot of them, I think, I don't think crane flies necessarily are up like
super high,
like high or anything. I think they're more like around the vegetation and hanging out. So they're probably easy pickings, especially for like the poor whales, things like that. But that was really interesting. So I think, I think they're partitioning at least the birds and the bats a little bit. That's how they're
so they're doing it on this moth dividing line, or at least the eared moth.
At least the eared moth dividing line, yeah.
Ears with real serious air quotes.
Right.
It's a different kind of ear.
Right. Now with bats, it gets way more complicated. In fact, we're in the process of analyzing all that data right now. And I've come across papers where, based off of like their morphology and echolocation behavior, that bats actually do partition the vertical airspace, to where they're not like interfering with each other's echolocation and things like that. And then of course there's different foraging modes, right? You can put them into guilds, like, some bats kind of use an edge space where they, will hunt around like the 10 or 15 feet or whatever
around like a tree or a canopy or something.
Exactly.
Right.
Whereas like a hoary bat, for example, and you can see that reflected in their morphology and their echolocation behaviors. Whereas like a hoary bat is, really stout, long, like narrow wings, like high wing loading, high aspect ratio, meant to just like a
cover
like
ground.
A fast flyer, right?
Right.
And so it detects its prey from quite a ways away, and then just basically blitzkriegs it.
Right.
Whereas these other ones, have wider wings, shorter wings, they're more maneuverable. Then you get like trawling species, like I think the little brown bat is like a trawling where they literally just fly above like a river or calm water, and just like scooping up the midges that are emerging or whatever, things like that. And so there's some partitioning going on that way.
Well, even partitioning with the feeding buzzes, right? Because those feeding buzzes are only reflecting off of certain sizes. Like if you're too small or too big.
Exactly. And so their characteristic frequencies, I get. And I'm not a bad expert. I mean, I've been learning steep learning curve, and Nate's been teaching me a lot about this. But you'll have like, and that's how they identify bats with echo, right? It's like, they'll have like a characteristic frequency that's a certain kilohertz or whatever, a range, and that's kind of what they're using.
And you've gone out with the bat finder?
Oh yeah. And actually Nate's got me now where it's like this microphone that I put on like a weather stand, and I put it on a piece of conduit, and it's like, so when I go out to do moths,
it's recording all the feeding buzzes?
It's just recording everything, and it's like got a big box, and I just set it up and turn it on and give him the card at the end of the year.
Even from the house here, I went out, Tom Forwood had the state of Montana's one and only bat finder box.
Oh yeah.
And we went out just on the river here, and I think we ended up with like 6 species.
Yeah. It's amazing, isn't it?
It's like, oh yeah, that's that.
Yeah, those are really cool. I think the sonobat or whatever it's called, right? To tell you like in real time, spit it out. Yeah, this microphone Nate's got, it's like it's like big time. Like it records like continuously, and like it's highly sensitive and
data analysis of all the feeding buzzes.
I have no idea about
the only thing I know about bats. Well, I know a little bit of bats. My favorite is there's only in Montana there's only one audible bat.
Oh, really?
You can actually hear spotted bat.
Oh, okay.
So their feeding buzz is actually audible.
Oh, wow.
So if you ever go out like to the CMR on the Missouri River, and you get around spotted bat, you can actually hear them.
Oh, be darned. Let's keep our ear out for that. That's pretty cool.
Unless you're going deaf like me. Yeah. So you find this partitioning in prey species, which obviously, they might not be happening at different times of night, but they're at least segmenting themselves somehow.
Right. And I'm not, getting down to the natural history of some of these insects, like, what level of the vertical airspace are they predominantly flying and stuff like that. It can be challenging because there's just not a lot of like information known about it.
Right.
But that's one of the things we're looking at with our bat paper now is we broke it down into, so we actually measured, so we took the top 20 prey taxa across all the bats. Mm-hmm. And then I identified, we got those like taxonomically sort of like into like at least genus or whatever. And then I sent those, they call them bins, barcode index numbers. And so I sent those bins to the lab and they actually pulled specimens and measured the length, width, depth of them and stuff. And so we're including size, and we're also looking at like diel activity. So are they crepuscular, nocturnal, or diurnal?
When are they taking off?
Right, because then that can give you an idea of like, okay, if you get like a housefly, a muscid fly or musket fly in the diet, and it's diurnal, then they're probably, that bat is probably gleaning it in the evening, right? It's probably that fly.
Or they're catching it just like that little cusp.
That little cusp before,
like a big brown bat, you'll see those things out early.
Exactly, and so and then we're also looking at. So there's been a lot of speculation about like the biomechanical limitations of bat bite force and their dentiaries related to insect hardness. Mm-hmm. And I think like big brown bats are presumed to eat a lot of beetles and stuff, which we interestingly haven't found. Most of what we've found them eating is crane flies and moths, with some beetles.
Right.
So
now, is that because there's not a little? Well, there's always a lot of beetles. I mean, there's always a lot of beetles, but enough flying beetles, or
right. And it also could be like a primer issue. So this DNA barcode is not perfect, and so certain insect taxa, like they kind of use like a universal primer to amplify this DNA. And so certain taxa of insects maybe don't amplify as well as others.
That has to be refined.
So there could be a bias in the DNA sequence.
And there might have to be a new primer.
And there are new primers.
You run primer for this, you run another primer for that.
Exactly. And so there could be some of that going on. But so we're including that. And so I found some papers where folks actually used like scissors. They went and collected a bunch of insects and they actually used scissors to cut them latitudinally and longitudinally, and then measured like the force.
It took to go through
to cut through the and they basically, what this one paper found was that on average, so like for the same size beetle or moth, the beetle is 3 times harder than the same size moth, right? And so what we did was we took our size data, and then they found a really distinct relationship between insect length and hardness. So like the longer the bug, the harder it's going to be, or whatever, the harder it'll be chewed.
Well, it'd be the difference between like crusty bread and Wonder Bread.
Yeah, right.
Still, I could eat both, but
right.
if I'm going to eat it all day long, I'm going to go with the Wonder Bread.
Exactly. And so we sort of developed a ranking system of where like mayflies, for example, are like the softest.
Right.
And then like, in general, like Neuroptera, like the lacewings and moths and some of the flies, minus like the midges and mosquitoes, which might be more like into the mayfly category, are like similar hardness. And then your beetles are your hardest, right? But then there's a length relationship too, right? So there's certain soft-bodied beetles that they consider soft-bodied, right? And so we looked at our length data and all our measurements and stuff. And we kind
of grease beetles, where they have that real soft,
soft, and some of the like flower beetles and things like that. And so we came up with this ranking system of like 1 to 4, and then so we sort of ranked the prey. Hardness from 1 to 4, like 1 being the softest, 4 being the hardest. And then, so for example, like our largest beetle in the diet, I think, was a carabid beetle. It was like 15 millimeters long,
right?
Something like that.
It's a big honker.
But we also found some really large sphigids moths. So like this pachysphinx, it's called, and it's a really large moth. It's like 38 millimeters long, its body. And so that's a hummingbird size.
It's
a big moth.
With sphinx moth.
Yeah.
No, but that's like a hummingbird size moth.
It's a big moth. Yeah. And so according to this paper that we sort of based these things off, and they had an equation that you could like actually, type in and get but the moth would have to be 2.5 times the length of a beetle to be the same hardness. And so for like the Pachysphinx moth, I put it as like a 4, right? So it would be the same hardness
as a little tiny hard
as
beetle.
A 14 millimeter hard beetle, right? So,
right.
And other folks have looked at sclerotization, like, so what's the level of sclerotization? But when I started looking into the data of that, I was like, that's really kind of subjective, right? You know, like how there's not a lot of data that says, okay, this is like a highly sclerotized insect, and this is a lowly, you know what I mean? So anyway, we're incorporating the size, the diel activity, the hardness, and like its primary habitat, right? So like if it's like larval or like primarily like freshwater, and some of the natural history of the insects, and we're putting that sort of into a model to see if we can determine any of these fine scale, you know, is there any like these fine scale things that the bats are using to partition? So like, you know, our like Myotis bats that are really close related and similar hunting styles.
Right.
Is one like using a different foraging mode to get like softer, weak flying prey versus like, you know, another one's using a slightly different foraging mode and echo behavior to get something that's a little stronger flying and a little harder.
And that actually just might be distance from vegetation in the canopy or altitude.
Exactly. And you know, in another paper I read, there could be just simply that they're, to avoid competition, they forage in different areas. So they're foraging the same habitat, but one goes over here and one goes over here. You know, and in the summertime, there was one study done that showed that when insects are abundant in early summer, that the bats are actually competing more for sound space, they called it. Right. So it's that vertical partitioning. So because the prey is abundant, right? So they're not really necessarily competing for the prey. You guys take the
low level, we'll take the mid.
We'll take the mid.
Those guys will take the high. Above.
Exactly. So we're hoping to flesh some of that out with this, you know, this new study we're doing.
So that being said, we've talked a shit ton.
We've talked a lot. I talked your ear off.
So how do people reach out to you if they're interested in data and research? And
you bet. So we've got a website. It's nrres.org. We're also on Facebook, so you can look us up in Northern Rockies Research and Educational Services there. And, you know, we've got an email listed on the, you know, our website or on the Facebook. You have an option on the website to actually sign up for, you know, we try to do newsletters, things like that. So you can sign up to keep up to speed there. And then, you know, our Facebook page, we try to, you know, especially this time of year, I mean, the winter, it gets a little bit, we don't do a great job of like,
moths flying.
Right. So, but in the summer, you know, we're pretty good about, you know, posting about what we're doing and cool moths we're finding or whatever. But yeah, you could reach out that way. And yeah, and if you,
well, I have to ask you the operative question we ask everybody.
Yeah.
What's your spirit animal and why?
Well, you know, it traditionally was a snowy owl because it was sort of a symbol of this like sort of purity and strength and stoicism, you know, and just being, and not only that, but being sort of like, I guess kind of being like different than all the other owls, right? I mean, even though people think that they're diurnal, they're really not, but they're actually
crepuscular.
But they're actually up there on the tundra, you know. When I was up there, I mean, they're active like all day long kind of thing, you know. So they're sort of this outlier, but so the snowy owl was traditionally my spirit animal.
Snowy owl.
Yeah, but you know, I might have to go with a moth one of these days.
Sphinx moth?
A sphinx moth. Well, actually, you know, my favorite moth is called Berencia. It doesn't even have a common name. It's called, the shell-formed brocade, I think, actually, is the common name.
Let's stay with Berencia.
Yeah, so it's Berencia conchiformis, and it's a moth you can find around here locally in, like, May. And I call it the galaxy moth because it's got this, like, really cool, like nebulous white patches on its forewings, but then it's got this like iridescent green, like speckling all over its forewings. It's a really beautiful moth. So anyway,
there you go. Well, thank you, Matt.
Yep, thank you, Rad. I appreciate it.
All righty.